Okay, I'll now be going through the 2021 MCQ questions 11 onwards. Let's begin with question 11. Which graph shows the changes in the concentration of a product in the enzyme controlled reaction correctly? Okay, so we know that in a reaction, enzyme catalyzed reaction, you go from the reactant to the product. So as the reaction proceeds, you will get more and more products and less reactants. So actually graph A is the best answer because you see that the amount of product is increasing as the reaction goes by and eventually it reaches a plateau. Because if you started off with only a certain amount of reactants, you will get a fixed amount of product at the end. All the other options don't quite make sense. You shouldn't get less and less products. You also shouldn't get more products and suddenly get less products. Okay, and same here, you don't get a constant amount of product, then it dips and then it goes back to constant again. So A is the best answer. Let's go to 12, another enzyme question. The graph shows the progress of a reaction in the presence and absence of an enzyme, which shows the activation energy in the presence of the enzyme. First, you have to identify which is the activation energy, right? The activation energy is actually B of the original reaction without enzyme present, because I see that this is the energy that the reactant started off with, and this is the amount of energy that needs to be overcome before the reaction can proceed. That is the activation energy. What enzymes do is that they lower the activation energy, and we can see they lowers it by this much, such that the activation energy required is left at you only need A left. Okay, that's the amount of energy that you need after an enzyme is present. Sorry, not after, but when an enzyme is present. Okay, so A is the best answer. Let's go to 13. Question 13 is a question on comparison of prokaryotic and eukaryotic cells. They're asking you which combination is found in prokaryotic cell. The word prokaryote means that it does not come with a nucleus. Okay, no nucleus. But other than no nucleus, actually, what it really, if we broaden the idea of it, is that in prokaryotic cells, they have no membrane-bound organelle. Okay, there are no membrane-bound organelles. Means there's no endoplasmic reticulum, there's no Golgi apparatus, don't have at all. Okay, so which combination is found in a prokaryotic cell? You shouldn't find any nucleus. Can you find RNA and DNA? That's still possible. Okay, can you find ribosomes? Right? Ribosomes, yes, you will find ribosomes. Ribosomes are needed for protein synthesis. Okay? And ribosomes are not membrane-bound organelles. Instead, they are made up of a mixture of proteins and RNA. So C is the best answer. Next, let's go to question 14. Question 14 is a question on the membranous system inside the cell. So they say membranous sacs containing products of metabolism are formed by the endoplasmic reticulum. Membranous sacs, I think we are referring to vesicles. Alright, so where are these products transported to? Okay, why would you need to produce vesicles from the endoplasmic reticulum? Where would the products within the vesicles, okay, be transported to? Well, they can be transported out of the cell via exocytosis, right? Because we know that in exocytosis, vesicles containing substances can fuse to the membrane. And when they fuse to the membrane, what you find is that, okay, this is the vesicle that's fused to the membrane. Now the substance will be secreted out of the cell. Some of the vesicles can contain products that will remain in the cell. An example are lysosomes. Lysosomes contain hydrolytic enzymes that remain within the cell. So A is the best answer. It's not lysosomes only, it's not inside the cell only or outside the cell only. Actually, it's both inside and outside. Let's go to question 15. Shows a figure, shows an electron micrograph of an organelle. Which option correctly describes the processes taking place in the organelle? Okay, A. ADP is produced to provide energy. It's not true. You should change this to ATP. B. Lactic acid is produced by fermentation. This is not done inside the mitochondria. It's actually done in the cytoplasm. Okay, remember, glycolysis takes place in the cytoplasm, right? So it's not within the mitochondria. So B is also out. C, light is absorbed at the inner membrane. No, this is more referring to the chloroplast. And actually also for the chloroplast, it's not the inner membrane, it's the thylakoid membrane. So D is the best answer. Proteins are synthesized at the ribosomes. Do you know that inside mitochondria, there are also ribosomes? Mitochondria have their own ribosomes inside to synthesize proteins. Here you also see some ribosomes on the rough endoplasmic reticulum, right? But you also can find ribosomes inside the mitochondria. So D is the best answer. Let's go to question 16. The diagram represents two kinds of molecules found in the cell surface membrane. Which part affects fluidity of the cell surface membrane? But first and foremost, what is this and what is this? This is likely a phospholipid. How do I tell it's a phospholipid? Because this looks like the hydrophilic head, and these two look like the hydrophilic tails. This is likely some sort of a glycoprotein, because you can see that it's likely the protein part and this likely the sugar part. Okay, you can see these look like sugars. So which one affects the fluidity of the cell surface membrane? Glycoproteins, when this is embedded within a cell membrane, doesn't really affect the fluidity. However, the fatty acid chains of the phospholipid can affect the fluidity, depending on how saturated or unsaturated it is. The more kinks there are, the more fluid the cell surface membrane will be. Because if you have phospholipids of a cell surface membrane that are all saturated and there are no kinks at all, you'll find that they can pack very closely to each other. This makes the membrane more rigid. But the moment you have kinks in the phospholipid, you find that the phospholipids cannot pack as closely together. They now cannot pack as closely together. This makes them more fluid, right? So depending on how many kinks there are, it can affect the fluidity. Therefore, B is the answer. Let's go to question 17. Which structure is likely to form when phospholipids are mixed into water? If they're mixed into water, okay, so to answer this question, you must remember that there is a hydrophilic head and then there are hydrophobic tails. So what will make sense is that you need to find all the hydrophilic heads interacting with water. You find that A is possible because all these hydrophilic heads here are interacting with water. Hydrophilic head here also interacting with water. So A is possible. What about B? You see that the hydrophilic tails are interacting with water. This is not correct. Same here, this is not correct. Here, we see again the hydrophobic tails are interacting with water. This is not correct. This is still possible, hydrophilic heads interacting with the water, but because of this part, it's wrong. D, you see that hydrophilic heads interacting with water, correct? But the hydrophobic tail shouldn't be interacting with water. So D is also wrong. Therefore, A is still the best answer. Let's go to question 18. Which statement correctly describes osmosis? Okay, A. It cannot occur across the phospholipid bilayer without the presence of embedded aquaporins. Actually, it can. Alright, water can pass through the cell membrane, just that it happens at a slower rate. Why? Why can it happen? Because water molecules, they are small. But because they are also polar, that's what makes them pass through quite slowly. B. Osmosis can only occur if there's a partially permeable membrane. This is true. If there's no partially permeable membrane, you will not find a net movement of water molecules from one side to the other. Instead, you'll just see diffusion happens. You see diffusion of the salt or the sugar, but you will not see the a net movement of water molecules. So a membrane is actually very important. C. Water molecules move from a region of high water concentration to low water concentration. This is not true because we don't say water concentration. Concentration is used to describe the amount of things dissolved in water, like sugar or salts. Okay, water does not in itself have a concentration. Instead, you should be using the word potential. D. Water molecules move from a region of low water potential to high water potential. This is not true. It moves from high to low, not low to high. So D is also wrong. Question 19. a plant cell was placed in solution Y for 10 minutes, resulting in a plasmolysed plant cell. So what has happened? What has happened is that some substances, or rather some water, has left the cell, right, into solution Y. So what is found at Z? Okay, we find that Z, because some of the water has left the cell membrane of the cell will peel away from the cell wall. This is the cell wall, right? And then leaving us with this, in a sense, spaces. But this is not really empty space. It's not filled with air. Instead, we know that the cell wall is fully permeable. This means to say, solution Y that's right over here, it will just pass through the fully permeable cell wall into these spaces. So actually what is in these spaces is solution Y, right? And it's not solution X either, because it's only the water from solution X that's going out, not solution X in itself. So yes, solution Y may become more diluted when more water comes out, but it's still solution Y. Okay, and it's also not just air. Next, let's go to question 20. Question 20 shows a whisking tubing. And what you find in a whisking tubing, there's 2 m of sucrose. Outside of whisking tubing, 0.5. Okay, that means that we can say the inside of the tube is hypertonic. Hypertonic means there's more dissolved substances. Outside is hypotonic. That also means that inside the whisking tubing, the water potential is low. Outside, the water potential is high. That means you expect water to be moving in. So which diagram most accurately represents the movement of water molecules at region X, which is across the membrane? So if the small little white circles represent water, the large one represents sucrose, you should expect to see water molecules moving from outside to inside. So these arrows are correct, okay, option D. And you'll find that there's more sucrose on the inside compared to outside. So option D is the best answer. Okay, so there's questions 11 to 20.